Hello, everybody. My name is Kavya. I have been working in the Manning Lab for a little over a year now. I am an analyst and I have been working at BDC. I've been using a lot of biodigested catalysts powered by Terra, and I'm very excited to present my project to you all today, which is gene-environment interaction between state-level social determinants of health on the age of onset of venous thromboembolism. There are two main goals for this project. The first is to determine if there is a genetic basis for the age of onset of venous thromboembolism, or VTE. The next is to determine if there is gene-environment interaction between state-level social determinants of health on the age of onset of VTE. We used multiple resources to get our data for this project. The genetic data came from 1,535 samples with whole genome sequence data. The data source was NHLBI TOPMed, whole genome sequencing of venous thromboembolism, or WGS of VTE. We also had phenotype data. We had 338 VTE cases from an inception cohort of Olmsted County, Minnesota residents, with a first lifetime objectively diagnosed idiopathic VTE during the 40-year study period from 1966 to 2005. The data source was NHLBI TOPMed, WGS of VTE. We also had 1,197 VTE cases who were previously recruited as part of the Geneva study. This data source was from a different source. It was the National Human Genome Research Institute Geneva Genome-Wide Association study of venous thrombosis. Now, it's important to note in the phenotype data that we had from VTE, the NHLBI TOPMed, we had phenotypic data for the 338 participants, but we also had 1,197 participants from the Geneva study who did not have phenotype data. This is why we had to go looking for the Geneva study so we could find the phenotype data to merge with the dataset, which we'll talk about later. I want to talk a little bit about the ScHARe data. So the ScHARe data focus on U.S. chronic disease indicators, or CDI. There are 124 chronic disease indicators that are important to public health practice. Now, this ScHARe data repository focuses on uniformly defining, collecting, and reporting chronic disease data that are important to public health practice and available for states, territories, and large metropolitan areas. Examples of these data include mortality due to diabetes and asthma hospitalizations. For our purposes, we used the overall conditions category, and this category was related to current lack of health insurance among adults aged 18 to 64 years. We wanted to look at this specifically for our dataset. The CDI dataset is one of the many datasets available in the ScHARe data repository. So for this project, we used scripts and workflows. These are available as a public workspace on biodigested catalyst powered by Terra. The workspace name is Interoperability Showcase, NIMDH ScHARe and NHLBI biodigested catalyst. There is a link to the workspace provided in the presentation. As you can see here, we have the workspace with the dashboard, and we also have the workflows that we used and the notebooks that we used for this project. So here is the basic overview of our analysis plan. The first step was to prepare the genetic data. Here, we exported the whole genome sequence data in VCF format from biodigested catalyst powered by Gen3. Then we used the workflow on biodigested catalyst powered by Terra to filter the VCF file. And then we used workflow on biodigested catalyst powered by Terra to convert the VCF file to bgen file, which we would then use for our analysis. After preparing the genetic data, we would prepare the phenotype data. we would export the phenotype data from the biodata catalyst powered by PicShare, and then we used interactive analysis on biodata catalyst powered by Terra to access and process area-level data from the ScHARe portal and create the phenotype file. After that, we performed the analysis. We used the workflow on biodata catalyst powered by Terra to run the gene-environment interaction analysis, and after that, we used interactive analysis on biodata catalyst powered by Terra to summarize the gene-environment interaction analysis through Manhattan plots and QQ plots. I'll show images of the Manhattan plots later on. So to do the genetic data preparation, we exported whole genome sequence data in VCF format from biodata catalyst powered by Gen3. So after logging on with an Era Commons account, we would search Mayo VTE in the project ID search bar and select the TopMed Mayo VTE GRU. Then we'd select Freeze 9B in call set and VCF and TXT in data format. We need both VCF for the genetic data and TXT for the various maps and information about the data that were helpful when doing the project. There should be 32 files. After selecting all the files, you would then click Export All to Terra here. It'll be highlighted once you select the files. There are step-by-step instructions in this PDF that is provided on the slide, and the workspace files can be found in the Interoperability Showcase NIMDH ScHARe and NHLBI Biodata Catalyst Terra workspace. After exporting the files, check the Workspace Data tab for the files. There should be, you see the file names here. Oh no, go back. And there should be corresponding DIRS URIs to each file. The DIRS URI files are how you can get the VCF files into the workflow to convert them into bGen, which is what we do here. We use a workflow in biodata catalyst powered by Terra to filter the VCF file. We take the workflow that has been adapted from the XVCF WDL on DocStore. The input will be the VCF file of the genetic chromosome that you want to use. We need to configure it to only keep the genetic variants with the value pass in the filter column. The output should be a VCF file. You can see the output here, it's a VCF file. Next, we want to use the workflow in biodata catalyst powered by Terra to convert the VCF file to bGen file. bGen file is what we use in our genetic analysis tool. We use the workflow genotype conversion WDL on DocStore. The input should be a VCF file. The configuration should be conversion parameter is VCF to bGen. And the output is a bGen file. You can see this would be the input file, the array here, the VCF file. And you can find the output bGen files here. This is the bGen file, and this is a sample file. We want the bGen file. Then we would export the phenotype data from biodata catalyst powered by PicShare. This is important for the next step, which is preparing the phenotype data for analysis. We would access the PicShare controlled access data, logging in through Era Commons account, and select the Mayo VTE PHS001402 dataset. We would select all the variables in the dataset. There should be 338 participants. Export this file as a PFB, you can see here, and export to a Terra workspace. And then you should have the data dictionary and individual level data available in your workspace. Then you can create a notebook to prepare the phenotype data file for analysis. The notebook we have available in the workspace is called Mayo VTE Variables ScHARe Interoperability. And with this notebook, we can make a Mayo VTE for analysis TSV file. Now we need to combine Mayo VTE and Geneva phenotypes. This involves merging the datasets by the appropriate subject IDs. By merging them, we should be getting a dataset, and here is the table of that dataset, the descriptive table. The primary outcome for this study is age clot, with the age at time of blood clot, with a mean of 50.83 years, plus or minus 16.98 years, missing 3 people. This is the notebook prepared phenotype file for analysis. After that, we'd have to use interactive analysis on Bidata Catalyst powered by Terra to access and process area-level data from the ScHARe portal. We would navigate to the ScHARe portal. There's a ScHARe workspace you can see here. Workspaces ScHARe. Find the common disease indicators data dictionary, and resources. Oh, sorry, that should say chronic disease indicators. The notebook in the workspace that we were working with and that it's available for you to see is the chronic disease indicators ScHARe dataset R. Next, we want to start working with this phenotypic data from the, not phenotypic data, the data from the ScHARe portal. We want to make a dataset that is 1 row per state and save it to the workspace bucket. This is to be able to merge it with our phenotype data file. After making a dataset that is 1 row per state, which you can see here, saved as CDI per state.csv in the ScHARe CDI folder. This notebook is chronic disease indicators ScHARe dataset R. We can read in the CDI per state dataset in our notebook Mayo VTE variables ScHARe interoperability notebook and select only the columns necessary for analysis. We need the column with the location data and the variables that we want to use for our analysis. This variable is category overall conditions. Variable is current lack of health insurance among adults aged 18 to 64 years. And you can see here, we read the necessary libraries and make a subset of the data. After that, we have to merge the state level data with the phenotype data. So that way, we have this data for the different participants depending on what state they're from. We had saved this to the workspace files as Mayo VTE ScHARe analysis CSV file. After this, we want to perform the genetic analysis. So we'd use a workflow on Bidata Catalyst powered by Terra to run a gene environment interaction analysis. The workflow is a GEM workflow on DocStore. The input should be an array of bgen files, and we need the phenotype file, which is the file with Mayo VTE and ScHARe. The configuration should be outcome age clot. Exposure is OVC, OVC1, overall age-adjusted prevalence from 2011 to 2011. Covariates are reported sex, clot height CMS, which is the height at time of blood clot in centimeters, clot weight KGS, which is the weight at time of blood clot in kilograms. Then we have ethnicity and the different indicator race indicator variables, which are race African American, race American Indian, race Asian, race other, race white, and race Pacific Islander. These would be in the input of your workflow, and the output should be genetic summary statistics. the GEM results here, the text file. After getting the results file, the text file, we need to read in the summary association results and create plots. For our purposes, we created a Manhattan plot that you can see here. This notebook is the GEM results final models, and these results can be used to generate Manhattan plots, QQ plots. Specifically, we ran the analysis on chromosome 21, but as a model, but you can run the analysis on whatever chromosome you need.